Believe we are live. There's a tiny bit of delay here in this streaming situation, but we are live and we are here to what are we here to do? We're here to talk about science. So let's start a show. Oh, you're live. There we go. That's what I like to see. So let's start this show in 3, 2. This is TWIS. I am not on the right page. Gosh darn it. Ha! Try it over again. Kiki.
Just call this week's episode Technical Difficulties, you know?
We'll fix it. It's just going to happen. We're going to fix it all up. It's going to be fantastic. Perfect and wonderful. Beginning in 3, 2. This is TWIS. This Week in Science, episode number 734, recorded on Wednesday, August 14th, 2019. What's that noise? Hey everyone, I'm Dr. Kiki, and tonight we will fill your head with free will, sperm, and monkey worries. But first...
Disclaimer, disclaimer, disclaimer. Every once in a while, it is worth taking a step back from everything going on around you. Everything going on at work, everything going on in your home life, social life, everything going on in the news around the country and around the world. Set it all aside for a moment. It'll be there when you are done here. And take this moment. Ask yourself a question. If I could learn something new, what would I most like to learn about? And whatever comes to mind, whatever piques your interest, whatever subject occurs to you that you do not know as much as you would like to.
Can we pause for a second and turn your audio down? They're saying it's really loud.
Yeah, well, I don't have audio control separate from...
You have a mixer. Didn't we get you a mixer?
Yeah.
I got me a mixer, but...
We got you a mixer.
I got me a mixer, and I also, whatchamacallit, have had the same settings set forever. So it seems to be interacting with this thing a little bit different. Okay. How's this? Disclaimer, disclaimer, disclaimer.
No. Too loud.
I think that's just me. I don't even think
No,
it's
it's like,
microphone.
there's distortion.
Disclaimer, disclaimer, disclaimer. Is that good? Put the mic way over there.
But you should be closer to your mic. Why is it set so loud?
Disclaimer. It's not set loud. It's the same.
Is.
It's exactly the same. Nothing has touched that mixer.
But it wasn't this loud last week.
Nothing has touched the mixer. No, that's all I can say.
Just maybe in some of the unplugging and plugging that happened, a contact is less dirty or something like that.
Okay.
And I don't know.
All right, here, let me go turn down some gains.
Right, Mataguro is right. You got 2 mixers and you didn't get charged for one. Right. Something happened and we haven't heard a click yet. So who knows? Oh my goodness.
I mean, jeez.
I'm trying to figure out how to make this work better.
Sorry.
We will. Yeah, apologies for this not being wonderful and perfect. We'll get there.
Mic check 1 2 1 2 1 2.
I think that's better.
All right.
Mm-hmm.
I think when you go loud though, you got to get a little off center from the mic. But when you're quiet, you got to get in the mic. But yeah. Identity 4, hit the emergency stop button. Well, we'll do the emergency start. Identity 4, should we start from the top or start from the disclaimer? It's a bit better. Let's just start from there. Do that.
Disclaimer, disclaimer, disclaimer. Every once in a while, it is worth taking a step back from everything going on around you. Everything going on at work, everything going on in your home life, your social life, everything going on in the news around the country and around the world. Set it all aside for a moment. It'll be there when you are done here. Take this moment. Ask yourself a question. If I could learn something new, what would I most like to learn about? Whatever comes to mind, whatever piques your interest, whatever subject occurs to you that you do not know as much as you would like to, do some digging, some reading, Googling, YouTubing, or do some testing, some experiments. Feed that curiosity with a bit of research. The mindset of inquiry isn't limited to the lab bench or a massive data set. It is something you can take with you anywhere you go. And if you follow where it leads, you might just find yourself listening to another episode of This Week in Science. Coming up next.
Sorry, I was listening to your audio. Now music plays.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to discuss all the news we saw fit to bring to the show, which is a significant amount. There's been some fun news this last week, so I am absolutely looking forward to digging into some of it. So tonight on the show, I have stories about space dust, free will, and first cells.
The first cells?
Yeah.
What?
What do you have for us, Justin?
Oh my goodness. That's the list I'm trying to pull up because I was over here at this thing. What do I have? Okay, so I have underwater Neanderthals. I've got microplastic snow. What makes monkeys worry? And how to reignite a greener future with airships.
Oh, you've always been a fan of the airships. Didn't those fail back in the 2000s or something? Anyhow, let's have that conversation later. Blair, what's in the animal corner?
Oh my goodness. I have Goliath frogs. I have sperm. And I have daddy long legs.
I love daddy long legs. They're great. Okay, I'm glad you're taking care of those guys. Looking forward to the animal corner. Okay, let's jump into this show. I would love to remind you that if you are not yet subscribed to This Week in Science, you can do that. Hey, we're almost to 13,000 subscribers on YouTube right now. We're about 9 subscribers away at the time of this broadcast. So, hey, maybe help us hit that big lucky 13,000 in the next couple of hours. That would be really awesome. Also, you can find this podcast all places podcasts are found: Google, Stitcher, Spreaker, TuneIn, Spotify, Pandora, radio.com. But TWIS.org is really where you can find all things TWIS. Let's jump into the science. Okay, me running this show saying, let's jump into the science, doing these things. Is it spontaneous? Is it free will, or is it just a pre-programmed genetic series of modules, programs?
And then the program glitched.
Oh no.
So this is that question of free will. Is it the thing that you actually are doing, or is it the thing that is in your makeup and therefore you have no choice but to do a science podcast? I find it to be a little bit tricky to say that we would be doing a science podcast over the internet based on our genetic makeup in some way. Somehow I feel that might be going too far.
Maybe I'd be doing a science podcast. I mean, I don't know. But the words that I say at any particular moment, are they genetically programmed? When does free will come into play? When does it not? There's a research There's a research because
There
I
is
can
a
see
There's a
research.
a
research.
research.
A singular research.
Singular, that's right. A study that is out this week that I found really interesting out of the University of Utah. The researchers published in Cell Reports, and they used not just behavior experiments with mice, not just genetics, but also machine learning to identify modules of behavior within the actions of mice during spontaneous food searching exercises. And so they had this landscape that had sand with wells in it, and they trained the mice in this round arena to search around and try and find food. Initially, when they tried it out, the food was visible and they could learn the places where these wells were found. Then they were asked to come in and actually dig in the sand to search for their food. Digging in the sand, searching for their food was more of this. The food was not necessarily in the same place as it was in their previous experiences. So they had to figure things out. And the behavior they discovered was made up of a whole bunch of little tiny modules. And it took recording these search experiences and breaking them down into little tiny components of what the mice were doing. And they were able to tie a number of their behaviors to genetic modules as well. So genes that control particular behavioral actions in the brain, which is pretty interesting. The idea they used this data from their recordings and algorithms in their machine learning setup to look into this foraging behavior, which described what they say is 71 separate modules. And 55 of those could be explained. 55% of the mouse behavior could be explained by these behavioral modules. 45%.
55%?
Yeah. So it's a little more than half, which means it's better than chance. But 45% of the action, not by much, right? 45% of the actions could not be categorized or classified. Now it gets really interesting because another aspect of this study was to look at behaviors as the mice aged. So young mice, middle-aged mice, and old mice. They found that in young mice, these modules explained behavior much, much more often. And there's less reliance on this modular behavior as the mice age. So the idea here the implication here is that these mice are relying less on specific modules that are maybe instinctual in nature. And as they age through experience and learning, they are turning to more spontaneity. And so age and experience leads to more spontaneous behavior, eh? Right.
That's what I was thinking too while you were talking about that was, you're born a precocial baby, a baby that has to be able to run around pretty quickly after they're born. So they are given a certain amount of natural instincts to figure out how to survive and basically just how don't you die when you're a baby, right? And so as you grow up, you get to learn more nuance and never go out in the clearing. Okay, don't usually go out in the clearing, but maybe if the coast is clear, you can run out there real quick, right? So like this nuance can happen over time as you succeed in not dying. So like the instinct that you're born with is really essential to that beginning part. And then you can kind of allow this more detailed behavior to show up over time as you learn.
Yeah, and the final aspect to the study, which really did dig into the genetics that are related, they actually looked at a couple of different genetic strains related to a gene called MAGEL, MAGEL2, and this is related to autism. This gene is related to autistic behavior in humans, and they looked at how 2 copies, no copies, or 1 copy of the gene, altered expression of the behavior that's encoded by the gene itself. And they were able to really show that parental genes influenced the children. But more often, they influenced the children when they became adults, which I thought was very interesting. So the modules, this specific module really became, especially the interesting aspect when we think about, there's a process in genetics called imprinting, in which you have 2 copies of a gene. You can't have both copies of the gene active at the same time in our chromosomes. So one of them gets made quiet, gets turned off, and that's imprinting. One is on and one is made quiet. And they found that even though a mutated form of the gene may have been present in that quiescent imprinted chromosome, that it still had an effect on behavior. So the mutation, even though we thought it was quiet and not having any effect at all, it still was having an effect, which is kind of an interesting aspect to it. But the big take home here is, to some degree, we have instinctual genetic instructions: feeding behavior, search behavior, hunger, satiety, different things that propel behavioral programs forward. And when one behavioral program happens, others are more likely or less likely to occur following it. So these things are related, but they don't completely define our behavior.
Yeah, I would like to know where these genes are, too, because the first thing I kind of started to think about was dogs. There's dogs with tendencies, you have dogs with herding instincts, dogs that will be more likely to dig under a fence than another dog, and dogs that will likely bark at people more. All these sort of things that we know certain breeds of dogs, which I'm sure can be associated to their genetics, but I wonder if just because they have genes that could be associated to their breed, if they would be as inclined. If those are the genes that are actually in place. I'm wondering where these regions of genes are that they've focused on.
Yeah, the genes that they were able to tie it back to, that they were looking at, they were looking at the differences in their genes, and they were tying gene expression to behaviors. And that's what the machine learning algorithm was there to do. And because they were looking at such, it's feeding behavior, the search for food and feeding behavior. And so you have anxiety, reward, life preservation, hunger, satiety, attention, navigation, memory. So these genes are probably present all throughout the brain. Yeah. These are genes probably present in all sorts of cognitive areas throughout the brain, and also in just the very basal levels of the brain as well. So yeah, they're probably very widespread, which is one of the interesting aspects of this. It wasn't like a very focused study on one particular gene. It's like very widespread.
Yeah, if you think about dog breeds, there might be a dog that, like the bloodhound, that's really well known for their sense of smell. And so that has to do with the parts of the brain that handle smell. And then there's animals that are really good at hunting. And so that's a different part. And then the ones that are really bad at, picking up on emotional cues, that's a different part of the brain. So yeah, I think also the thing about dog breeds too, is they're really, 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 really closely related. So they had to do quite a bit of selective breeding to get that happening. So it's probably pretty widespread changes.
And it's also a good point, too, because I would assume that these mice are pretty genetically similar. Usually they will do a study on mice where they are using mice that are, cut from the same genetic cloth. So when they're looking at expression in this case, they are kind of looking at really finite differences in what genes are turned on and off between these different mice. So that's exciting.
Yeah. Yeah, it is very exciting. But yeah, for now, don't let anyone say that we don't have free will. And there's a bit of programming there, but it's not all programmed.
You just get more with age.
Yeah, you get more unique with age, get more free will with age. There we go. Moving on to my other story. This one is a very interesting story. Ed Young at The Atlantic wrote up an article about this. But researchers at University of Washington have been looking at the beginnings of life. When life first began, we had just molecules bumping into each other, chemical reactions. At some point, cells became a thing. To create a cell, you need a membrane. But the oceans at the time were incredibly salty. All sorts of, natural salts, magnesium salts, and others that would destabilize fatty acids and their ability to create membranes. And so researchers have been trying to figure out how fatty acid membranes could have gotten their start if the salts are destabilizing them all over the place. How could this have ever happened? So these researchers at UW, publishing in Proceedings of the National Academy of Sciences this week, they started looking at the question of what about interactions with fatty acids and other molecules. There was some data that suggested that fatty acids start to interact with RNA molecules and that there's some stabilization that occurred as a result of that. And so the researchers asked, well, what about amino acids? What about these very, very basic building blocks of life, the stuff that came before RNA, the stuff that makes up the RNA, the DNA, the instructions that make us who we are? So they went further back and they in a dish full of salty water, they threw fatty acids and amino acids together and looked at what happened and discovered, lo and behold, there's an interaction in which amino acids stabilize the fatty acids and allow the formation of membranes. The fatty acids and amino acids create membranous structures, and not just single membranes where it's like a beach ball filled with air, but in this case, it would be a fatty acid spherical membrane with just salty fluid inside. No. Multiple membranes. So, like layers of an onion, membranes inside of membranes, which suggests that vesicles were even a part. These intracellular organelles could have gotten their start in smaller environments inside of the larger cell environments from the very beginning. So amino acids, the stuff that makes up life that was just floating around before RNA, stabilized fatty acids and could have created the membranes that created the first cells.
Wow. So then we don't even need the hydrothermal vent clay encapsulation at that point. You can already have a cell membrane.
Yeah, so they say that this isn't an either or kind of situation, and they are now going to move forward to looking in these different environments for specific facets of this that may have led to particular kinds of cellular evolution in the hydrothermal vents or in hot acid hot springs. So there are lots of different places where life is proposed to have gotten its start because of these extreme conditions with the chemistry that happens in these places. And so now that they have this basic data on how these membranes are forming, now they're going to go look and see how they could have formed maybe differently, or if there were different aspects of that formation in these different locations.
But still exciting that they did it in a pretty unextreme sounding environment. I mean, this is just components and salt water in a dish. There you go. Boom.
Yeah.
We have cell walls.
Cell walls. Ta-da! We got membranes for you. Lots of membranes.
That's amazing.
Have your cells, and there will be life. Yeah. Super excited. And this I think is like one of those findings that it's such a basic finding. It's not highly complicated. It's like, wow, these things, we didn't realize this before. They interact chemically in this particular way, and this is what happens, and you can see how the dominoes would fall as a result.
Well, yeah, and now you have a workspace. Now you have a place for, things to start combining and proliferating. And you have a lab in a protected space that can oh my gosh, it can happen over and over and over and over and over and over and over again.
Yeah. Yeah.
Which then also means if this is all taking place at the same time, it's fine, but that means that the origins of life could be multifaceted and not a single, I mean, chances being, but still not a single instance of and then starting, but could have been a much larger, grander experiment at the beginning.
Yeah.
That's fascinating.
Yeah, and then we look and there I was just as you're talking, I was thinking to myself, "Oh, I wonder if we've found fatty acids other places." And in March of 2015, NASA's Curiosity rover detected what was possibly a fatty acid molecule on Mars. So, fatty acids on Mars, where are those amino acids? We've got
See, there
we've
you go,
got the stuff.
panspermia. It's a thing.
Yeah. Are there other fatty acids that we've found in outer space?
Are those our fatty
Yeah.
acids, or do we have their fatty acids? Or are
Right?
they separate?
Are they separate? Yeah, that's it. Anyway, this very exciting, interesting questions. Cells and life. Okay, this is This Week in Science. Justin, what do you have there?
Oh, let's see. What have I got? Okay, let's dive into Neander news. There's a study published in PLOS One by Eric Trinkaus of Washington University. He was examining ear canals in the remains of 77 ancient humans, and they found external auditory exostosis. What? Yes, they found
What?
external auditory exostosis was common in Neanderthals. Oh, you're not familiar? Okay.
What is that?
Translation?
External auditory exostosis is a dense, bony growth that protrude into the ear canal, which in modern humans is commonly called swimmer's ear.
Ah.
And it is thought to be correlated, or is known to be correlated, with habitual exposure to cold water and chilly air. So such exotoses have been noticed in ancient humans before, but this is the first time they've really looked at the numbers and tried to figure out how that might have come about. So in the study, they looked at ancient humans, including Neanderthals, early modern humans from the middle to late Pleistocene epoch of Western Eurasia, while early modern human samples exhibited about the same frequency of the swimmer's ear as modern humans would likely have. It was exceptionally common in Neanderthals. I think it was more than half of them even had this. So they suggest that might mean that Neanderthals spent a significant amount of time collecting resources in aquatic settings. So they might have been fish diving in rivers and lakes.
With limited access to Q-tips.
With no Q-tips and a lot of the, banging on the side of the head to try to get the water. I don't know what. I've never had.
And no earmuffs.
I mean, really, were they fish diving in lakes, or is it just, was it just cold?
Yeah, was it just rainy?
Yeah.
Right.
I associate Neanderthals with cold climate. So it's just maybe it was just because it was cold and the bone protruded to keep the warmth in the brain, in the ear canal.
Right. So they what was interesting about this is amongst the Neanderthals, it was a more modest level among high latitude earlier humans, indicating higher frequency of aquatic resource exploitation. So they had a pretty decent range of sample size, and it kind of suggested it wasn't correlated with cooler conditions or cooler climates. That was one of the things that they kind of like you immediately jumped to. It's also what they kind of expected they would find, but it didn't correlate. So that's why they think it's quite possible that there was some fish diving going on in ancient Neanderthals.
Now we have to look for their flippers. No.
Or the scuba tanks. Where are those?
Next story.
I don't know. I'm just imagining. I mean, we really think of Neanderthals as these, big lumbering, kind of beefy people who I mean, maybe with stocky legs and kind of, the big forehead and everything, but I haven't really thought of them as swimmers. Now I'm going to have
So
to
Agile enough to
reinvent.
just reach in and grab a fish.
Right,
So
the
a couple-
agility would be yeah.
Yeah.
a couple of things to those points. There are islands. I think it's Crete, maybe. Some Greek island where they have found Neanderthals, which they know that the Neanderthals must. Well, one of the theories is they might have had boats of some flotation device to get across to these islands because they never would have been close enough to swim, at least not by a modern human. One of the interesting things about Neanderthals is they have these barrel chests. They have larger lung capacity. So they may have been actually, we've got a minute or so that we can go down and under. They may actually have been able to stay under longer than a modern human and, have better chance at grabbing a fish.
Well,
So-
why are they assuming fish? Why aren't they assuming it's the things that are more sessile, like oysters and clams and crabs and stuff like that actually be super easy to just dive down and grab?
It could be, and a lot. There have been some Neanderthal sites that were kind of close to oceans, but a lot of the times they're kind of further away from the ocean. But that's,
Oh, so you're saying it's fresh? You think it's freshwater, but there's freshwater crustaceans too.
There are, and also, it's another one of the problems with any sort of archaeology going into the past. Sea levels are so much higher now than they were. That those sites that we would have the best evidence of that, we need marine archaeologists to go out and find sites. That's a whole other problem. Hey, if you are a monkey, not
I'm
saying
a primate.
No, but if you were a monkey,
Okay.
If
Yeah.
if you happen to be a monkey, you might have a tendency to worry about things, predation pressures, resource access anxieties, occasional fear of heights. Maybe I don't know if monkeys ever get afraid of heights. But the thing that will make you worry the most is a molecule. Boosting a single molecule in the brain can change dispositional anxiety, the tendency to perceive many situations as threatening. In primates, researchers from the University of California at Davis and the University of Wisconsin-Madison have found the molecule neurotrophin 3 stimulates neurons to grow and make new connections. Findings here, they think, will provide new strategies for focusing on people with anxiety disorders. This is quotey voice of Andrew Fox, who's an assistant professor at UC Davis. There are millions of people worldwide who suffer from debilitating anxiety and depressive orders. These disorders are also some of the leading causes of disability and days lost to disability. So, in looking at this, they found that they can identify even. Oh, okay. So here, we'll go back. So this anxiety disorder often emerges around adolescence, can continue beyond that, stays with people their whole lives. They can identify children who display extremely anxious or inhibited temperament, and these people then are at greater risk of developing stress-related psychopathologies. So the roots of the study came from research done by a group about 8 years ago in pre-adolescent rhesus macaques. And researchers got their first glimpse of molecular alterations in the dorsal amygdala. They started to get a clue. So they speculated that altered processes in this region might underlie the anxiety issues. So since then, they've then sequenced RNA from the dorsal amygdala and identified molecules related to the anxiety. They eventually narrowed that whole pool down to this one molecule, which is a neurotrophin 3, which is a growth factor. So out of everything, they altered a virus to boost levels of neurotrophin 3 in the amygdala of juvenile rhesus macaques. They found that the increase in the neurotrophin 3 in the dorsal amygdala led to a decrease in anxiety-related behaviors, particularly behaviors associated with inhibition. So subsequently, brain imaging studies of the animals found that it changed activity throughout the distributed brain regions. that contribute to anxiety. So this one molecule is sort of like the catalyst for preventing, I guess, the anxiety from taking place. So it's really fascinating. I mean, you were talking about free will and how much we have control over our thought processes, and it could come down to whether or not this one molecule is being produced in sufficient quantities in your brain that will then have a cascading effect throughout the rest of the regions of your mind, and therefore your entire interaction to the world could be determined by just this one molecule. Really fascinating.
It's very cool. Take it out. Take my whole amygdala. I don't care.
No. I think you want this molecule, I think, is the thing.
Oh, you want
anxiety is it is in some amounts helpful.
It's beneficial,
right? It's
sure.
beneficial. It can be beneficial. You want to be nervous about some things a little bit. So you want to have that little bit of nervousness so you'll see that predator, so that you won't fall down the cliff, so that, there's certain things that are worth the anxiety. But when we talk about anxiety in the modern context, it often it can lead to these cascading effects that result in panic attacks and generalized anxiety that make modern life really hard to live. And you're like, I can't go to the grocery store. Okay. Or you go to the grocery store and you go, I can't decide what I want.
Okay. I know,
so it's useful. You don't want to get rid of it, but can you moderate it? Modulate.
There's something troubling about that version of the anxiety, because I get it in the ancients when I really can't go and take down a mastodon. It's just I'm really nervous about it. Like, it made sense that you wouldn't want to go spear a wildebeest even, but the grocery store, it's not going to. You're not going to get tusked or trampled.
Right, but that's where it goes wrong.
Well, and then there's all these sort of runaway adaptations or things that we want to adjust that, thousands of years ago wouldn't have made it to adulthood. And I feel like, I wouldn't have made it with my vision, for example. My vision is so terrible without contacts or glasses. I never would have made it to adulthood in the olden days. So there's lots of things that, thank goodness, we have found ways to fix and adjust and allow for people to survive through. And I think intense anxiety is one of those things. There's a certain level of anxiety that would have been completely a stopper in the past without proper medications or support systems or therapy or counseling. And so I think there would be a situation where you would have anxiety so bad that you couldn't take down the mammoth. And that was it for you because you couldn't get food.
Right. I don't know. As someone who suffers from anxiety, generalized anxiety that often leads to depression, if they can use this molecule to improve treatments for it, I honestly can say it would improve the quality of my life. And yes, please. I want to go to the grocery store without freaking out.
You don't want to wake up at 2 a.m, start thinking about things and spin out completely? Because I've done that too.
Yep. Yeah, it can be debilitating at times. And so, this is to find a molecule that is crucially incorporated into how anxiety progresses, whether it progresses, like that. It's an amazing target for future therapies.
I bet I have a ton of this. I am not nearly as nervous or anxious as I should.
Just a cool guy.
No, like even even when things are really going terrible, I'm like, oh look, things are going terrible. That's weird.
Just keep
I really don't freak out like I should. I should be more. I should probably be much more anxious at any given moment.
No, see, there's a positive word for that you could use like in an interview, and it's called even keeled.
There we go. My neuromolecules allow me to be.
I'm also blind to the.
See,
I'm also blind to risk and danger. I don't know if that's a problem.
See, you're even keeled. We're task oriented, which really just means we fixate on a thing.
What's going on there? I gotta know what's happening there. Gotta go.
Yeah,
Gotta,
yeah.
Okay.
Yeah.
All right. Well, you know what I'm fixated on right now? Blair's Animal Corner.
Yes.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh my goodness. I love this where we can see everybody while we're dancing too. It's so fun. Okay. On the Animal Corner today, I have a very exciting story about the world's largest frog. He made an appearance on the calendar this year. It's called the Goliath frog. These frogs are over a foot long. They can weigh 7 pounds. They are large frogs. And there's a new
7
study.
pounds?
Yes. There's a new study that indicates why they might be so large. It's not for defense. It's to be good parents. So this is a study in the Journal of Natural History that came out this week all about nest building in the Goliath frog. This is the first example of nest building in an African amphibian ever seen. And they have been observed in the course of this study moving rocks that weigh up to 2 kilograms, that's around 4.5 pounds, while building ponds for their young. So these guys, they're an endangered species. Their numbers have declined by more than 50% in the last 10 years, mostly due to overhunting and deforestation. These guys live deep in the rainforest. And the way it works is they are on these really fast cascading waterfall or like whitewater rapid type areas is where they lay their eggs. But you can imagine if you laid your eggs at the top of a waterfall, they wouldn't last very long. They'd get pushed right off the edge. Similarly, with a fast-flowing river, if you laid your eggs in that fast-flowing river, it would get pulled downstream real fast. Oh. Subsequent tadpoles have to have a place that's safe where they'll stay in kind of one spot away from the torrential waters, and also where they're protected from predators, because when they're eggs, they're absolutely sitting ducks. Not to be confusing with the different sitting frogs. I don't know. And then when they're tadpoles, they're similarly kind of helpless. They can eat and they can swim around, but they can't leave the pond. They're completely stuck. So the placement for a pond for eggs to go is actually really important to parental care. When we think about frogs and how they lay their eggs and leave, a lot of frog species, that's actually a lot of parental care, because a lot of work goes into the space where they lay the eggs before they leave them. So in this study, they identified 22 potential breeding sites. 14 contained nearly 3,000 eggs each. And they watched these nests, just normal people hanging out. The ones that showed the most recent signs of activity, they put camera traps down. They recorded time-lapse video using a camera trap. So they found that Goliath frogs create 3 different types of ponds. One is they'll clear naturally occurring rock pools of leaf litter and debris. So they just take like, oh, this is some nice still water. Let's pull out the debris and lay their eggs there. Okay. Second, they find a puddle, which has thick layers of leaves, debris, and gravel. So then it's a different kind. This is like the first one is like rock bottom. This one has a debris bottom. The one with the debris bottom, they dig out the leaf litter and gravel and push it to the edges, which creates a dam. So that slows down the water. And then the third type, they actually clear depressions of shallow water where there's already really quickly running water, and they move large stones, they move them to the edge, and they use the large stones like a dam, creating a circular pond. So
that's I mean, these are frog engineers.
Yes.
That's not just simple behavior. It's not like if it was one behavior, It's an instinctual learned thing. You just do the one thing.
No,
But
they like
they're
look around and go,
doing-
what would make a
Yeah.
good pond?
Yeah.
And then
Oh,
based on
that's
what they
brilliant.
find, they have these different methods. Absolutely.
Wow.
And so the dams, the third time, the dams with the large stone walls was the most reliable type. The eggs were least likely to spill out or become flooded through heavy rain. But where this gets really interesting is that in their time lapse, they also found that adults spent the night guarding the nest from predators. They left just before dawn. The scientists were unable to determine which sex was responsible for building ponds and which was for guarding. But part of how they found these spots was by talking to the hunters. Now, remember, this is an endangered species. One of the reasons they're endangered is from overhunting. But by talking to the hunters in the area, they were able to figure out exactly where to go. So this is a great example of maybe not wanting to alienate the hunters of a species immediately. Because they might actually have some really valuable conservation information. So they were able to find out where these frogs were. Now, one of the hunters that they interviewed described their behaviors in detail before they were able to record them. And this hunter suggested that males do the construction while females are the guards. Now, let me just say, there is a slight size difference in this species between males and females, but overall, they look practically identical. So I'm not sure how this hunter came up with this idea.
Because he killed it and ate it afterwards.
But how would he know if it was a
cut it
male
up.
or a female?
I don't know.
Remember, frogs don't have external girl parts and boy parts. So
he took an anatomy class and learned the difference between internal anatomy between males and females.
Yeah.
Oh, yeah, he did a frog dissection, maybe.
Right. I mean, if the hunter is hunting and eating the frogs, they could be opening them up. And, very often with fish, even, you don't know until you open up the fish and then you see, oh, they've got these eggs, they've got this is different. And you're still going to eat the fish. And it's just something that you see as you go through it.
Yeah, that's a good point. That's possible. Yeah, that would make sense. So Dr. Mark Oliver Rodell, who's the project leader and the president of Frogs and Friends, which I think I gotta hunt down and join, says,
absolutely,
the fact that we've only just discovered these behaviors shows how little we know about even some of the most spectacular creatures on our planet. We hope that our findings, combined with further ongoing research, will improve our understanding of the needs of the Goliath frog so we can support its continued survival. So there you go, big frog with a big mystery. So they think, this is the crazy thing though, is that they think now that the main evolutionary driver for them to get so large is to move these giant rocks.
I gotta be big and move the rocks. Move the rocks, protect my babies. Here we go.
Yeah. So I think that's so insane that they think they may have gotten to be these enormous amphibians, largest frogs on the planet, to build nests for their babies.
So that is a good hypothesis. The other part of it I would think of is if they're living in these fast-moving waters through near waterfalls, through evolution,
just sheer heft would be helpful.
The heavy ones stuck around, and if you were born the runt of the litter, you went over the fall. That was you just didn't have the weight to stay down, and so through natural selection, you should end up with bigger frogs.
It could be a chicken and egg or frog and egg scenario, right? Where where they could have gotten bigger in order to not fall off the falls. And then we're suddenly able to move things better. And then we're able to make
better.
Or they could have gotten bigger to make their ponds, to have babies, and then also were spit off the end of the waterfall less off. It could have gone either way.
Yeah.
But it definitely seems advantageous. So.
And it seems like more studies are to come to tell us more about this. I mean, it's just opening up questions, right?
Yeah, absolutely. And speaking of questions about raising babies, let me just ask you both. I know you the answer you have to say to this question, but you both have babies. Both have children. So I'm going to ask you, if you could have picked if your child was male or female, would you have?
No.
No.
Okay, now give me the real answer.
No.
No.
Okay, good. Just checking. Actually, I feel the same way. I asked my partner the same question. He had the exact same answer. But then I asked, okay, after the first one, or like if I had twin boys, would you want to pick
Okay.
the sex of the next one?
So
And that
hang
answer
on.
was, maybe.
So not to get too woke on you, Blair. But that even might not be your choice.
Gender might not be my choice, but in this case, sex could be your choice, which is what this story is about. And those are two very different things. So gender is the identity of the individual, and sex is the anatomy that you had at birth. So in this case, the sex of an individual is determined by the X or the Y that you get from sperm, from your dad. Your dad is completely the inadvertent decider in the sex of the baby when it comes out. But that could change. So in the latest, most successful attempt to select X sperm and Y sperm, this is from Hiroshima University, they were able to find a chemical treatment that could segregate specifically X-bearing sperm out from the Y-bearing sperm. This was performed in mice, but could technically likely be adapted to other mammals. They found that a chemical that bound to the receptors of X-bearing sperm slowed motility without impairing fertilization or viability. They also showed that they could remove this chemical afterwards from the sperm, and that then they could go on and be normal sperm. So when they treated the sperm with this chemical, this kind of X-selecting chemical, they were able to slow down all of the X sperm. They were able to take what was left, was able to move forward, pass through, and those litters were 90% male in these mice. When they selected the slower swimmers, the back end of the vial, if you will, the litters were 81% female. So it appears to work pretty well. It's definitely the most effective so far that they've been able to find. And this is capitalizing on the fact that the X-bearing sperm has a lot of genes that the Y-bearing sperm do not, because that's that whole extra tail right there, the X versus the Y. And so they were able to identify a chemical that specifically focused on those that slowed them down. Now, I'm talking about being able to pick your own babies. That's not really what the most likely use of this would be. Obviously, that's kind of the science fiction end result that we can talk about with this. And I'm sure there are some people that would like to make that decision, like people that have like 6 boys and finally want that girl, or vice versa. But the really cool thing from my perspective, thinking about endangered species, is if you only have a very small number of individuals left and you know what genetic stock needs to be expressed to keep that population viable, and so you really, 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 really need a female to go with the males that are currently in your collection, that's something that you could engineer. And I think that that's where this gets really interesting. There's also the whole issue of the fact that in labs, we found out that a lot of the lab mice have been male, right? So then you could try to engineer it so that you get a certain number of females so that you could have an unbiased group of mice in your lab, so you don't have this gender bias. There's all sorts of things that you could potentially do, scientifically speaking, with this technology.
Yeah, it seems a really interesting technology that it would have a lot of uses besides just allowing China to have all of the male children and none of the
Oh my
girl
goodness.
children, right?
Actually, they
See, the
probably
problem is-
select for girls at this point. I
At
think
this point,
I
Yeah.
think they've realized, oh, this has been a mistake.
Somebody was like, hey, it turns out you need both.
You do. You need.
So
you
the
need
way it
both.
works is, well,
Yeah.
yeah.
Turns out you kind of need both to continue to make more people.
Yeah, it's interesting though how this molecule, how they've been able to find this receptor, this molecule that affects the speed of the sperm, right? So not only can you could potentially in a vial be able to use the molecule to go, hey, that's a male or that's a female, but you could potentially use activation to the receptor to actually activate it to turn up the juice on slow swimming
Mm.
sperm
I'm
potentially.
also thinking if it's something that you could, this is like way into science fiction now, but
Yeah.
if you can identify this chemical and find a way to safely pass it through the digestive tract of an animal or a human, they could just, you wouldn't have to take it out and do this in a vial and then do everything in vitro. You could
Mhm.
potentially find a way to just through ingesting or injecting, you could give this chemical to an individual that would slow down the X sperm. Like if you really wanted Y babies, for example, if you really needed males, you could do that. And then in theory, during just normal copulation, in theory, the male sperm would reach the egg first.
I wonder how this could potentially influence men who have slow sperm or sperm that have motility problems, where that can potentially lead to infertility. And not just sperm count is a factor related to fertility. The motility of the sperm themselves is important. So, is this something that could be used to improve that factor in some people?
Yeah,
It's
good
another that's
question.
another it's another
Fast.
possible application.
Fast or slow, a large or small population. Everybody listening, just about. You are the most successful sperm at one point in your life.
It's true.
It is true. You were so successful. You made it. You
did
made it
it.
here.
You really accomplished the biggest achievement of your entire life before you were ever.
I
even-
exist.
There we go. Life. I exist. And this show exists. But only because of you. So let's take a quick break, everyone. We'll be back in just a few moments for more This Week in Science.
Can't explain the things you've heard with more than intuition. A lot of research shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I.
Thank you for being a part of TWIS. Thank you for joining us for this episode, and I hope that you are enjoying it. In the meantime, I did mention that, survival, it is the big thing, and TWIS, our survival, does rely on you. You are important to keeping this show going, and there are many ways that you can be involved in keeping this show going, in being a part of the community and enjoying what we do, learning about science, like we enjoy doing week after week after week. So let me tell you about a couple things. First, TWIS.org. That's where you want to go. TWIS.org is the home of all things This Week in Science. If you head over there, you will find our wonderful newsletter. There's a pop-up if you haven't visited the website before that will allow you to subscribe to our newsletter. We just sent one out today, actually. Right now, we're on a monthly schedule. It may increase with time, but if you are interested in getting our newsletter, be sure to check out that subscription form. Then, while at twist.org, you can find all the wonderful things like our Zazzle store, which is full of lots of goodies related to TWIS. We have all sorts of things, t-shirts, we have tote bags, which somebody in the YouTube chat room, Gaurav, is saying that they love uh, the tote bag, that it's great quality. So that's really wonderful. If you need a new tote bag for your groceries, maybe consider getting one from our Zazzle store with the TWIS logo all over it. Other merchandise has images from previous Blair's Animal Corner calendars, which she's working on right now. Did you know there's a new calendar coming out again this year? Stay tuned for that. We'll be announcing that within the next month or so. Then back to This Week in Science, we also have our Patreon community where you can click on the Patreon link at twist.org. It will take you to Patreon where you can click on the red button to become a patron. Become a patron at a monthly amount of your choosing. 3.14 is our pie level if you want to start with giving us some pie every month. Or at $10 a month, we will thank you, and above, we will thank you by name at the end of the show. If you want one of our new neat TWIS shirts, you're going to have to get into the neutrino support level at $25 a month. All this support helps to keep This Week in Science going and helps us do things like buy new computers and maybe new audio cables. A new audio cable for Justin. We might have to do that. We also have a donate button if you're not into Patreon. There's a PayPal button. That's right. You can donate using PayPal. And if you are not subscribed yet to the show, or you know somebody who's not subscribed, take them over to this subscribe button. Get them to subscribe in one of the ways that works best for them. Google Play, iTunes, or YouTube. Like I said, we are close to that 13,000 number. Let us hit that benchmark, and we're going to keep climbing, but be a part of getting us to 13,000 if you can. That would be wonderful. Everything you do for us, honestly, you help so much, and we really could not do what we do without you. Thank you for your support.
Can't explain the things you've heard with more than intuition. A line of reason shows the way.
And we're back with more This Week in Science.
Yes, we are. We're back again. And now that we're back, what do we have for us? Oh, it looks as though we have a This Week in What Has Science Done For Me lately? What has it done? This was a late, wonderful get in under the line entry. Thank you, Kristen, for writing in and for heeding Blair's call that we needed a What Has Science Done For Me lately this week. Kristen wrote in and said, I've struggled with getting pregnant for almost 2 years, so I often find myself feeling frustrated with my body and cursing human biology in the seemingly haphazard way that many lucky couples are able to conceive. But recently, I've started fertility treatment and learning about all of the incredible medical advances and options for couples like us has been pretty awesome. It feels super encouraging knowing that some really smart people are working hard to develop methods to help me and tons of other women start a family. Science gives me hope. Thank you, Kristen. And I'm glad you do have hope. I'm glad science is able to give that to you. And I hope to many other women and men as well, to partners around the world who are trying to conceive. Everyone, we need to continue filling this segment of the show with tidbits from you. What has science done for you lately? You driving your car right now. You driving a Tesla? Science behind that? You sitting at your cafe drinking your coffee. Is there some science behind that? Hey, everyone, you can write us a poem. You can send us a note. You can I don't know. There's all sorts of ways you can tell us what science has done for you lately. But the two ways these messages get to me, I'm just breaking microphones over here. The two ways these messages get to me are through one, a Facebook message on our This Week in Science page on Facebook. So go like our Facebook page and send me a message, and I will get that. Two, send me an email, Kirsten, Kirsten, at thisweekinscience.com. What has science done for me lately? And you'll get in the queue. And we will read your story. We will read your note. We will read your sonnet. We will maybe try to sing your song. However it works. All right, Justin, tell me some sciencey goodness.
It's a bird. It's a plane. It's a flying car. No. It's an airship. What? So airships, dirigibles, is that what they're called?
Dirigibles.
Like the Zeppelins, kind of like a blimp, or like Hindenburg. There's some talk, a study by the IASA led a study that suggests reintroducing airships into the world's transportation mix could contribute to lowering carbon emissions. And also could play an interesting role in establishing a sustainable hydrogen-based economy. So the airships were introduced in the first half of the last century, before airplanes were being used for long-range transport of cargo and passengers. Their use was quickly discontinued for a number of reasons, most dramatically after the Hindenburg explosion highlighted the risks. Yeah. And they have lower speed compared to that of airplanes. And back then, they didn't have the best weather forecasts. So you would go up on a sunny day and then quickly find a storm.
Lightning? Yeah, that's not good.
So we've had some considerable advances in material sciences, which this is, I think, in reference to the Hindenburg, which they believe was a coating on the outside that was more flammable than perhaps that they had realized. Our ability to do better predictive forecasts of weather, and also coupled with an urgency of reducing CO2 emissions. So transport sector is currently responsible for around 25% of global CO2 emissions caused by humans. And they're saying, oh, it's not a lot, but it's 3% come from cargo ships. But that figure is actually expected to rise. It's supposed to go up 2.5-fold, I suppose, until 2050. So it's an interesting proposition just because it would be really awesome to see airships in the skies, I think. They can also fly to incredible heights. I don't know. Most planes don't actually go up nearly as high as a balloon, like a flotation-y type thing. But they can get up there into the 80,000 feet if they want to. But what they're looking at is actually using hydrogen as part of the thing that they could be moving about. So you could jump into, you can load it with cargo. They can carry a tremendous amount of cargo. I think one of the last projections, the thing was as big as a football field, but it was something like 50 tons of cargo, something like that. They could put that up into the jet stream, and with very low fuel requirements, it could make it across the Atlantic. Right? And once it got there, it could unload cargo. Plus, it could also reduce the hydrogen on board by as much as 60, even 80%. And with that, because it's not carrying cargo on the way back, that would be sufficient to get it home again. So you could both be doing transportation of the cargo and hydrogen from place to place, and utilize as part of the hydrogen economy. So it's an interesting proposition. Mostly just because, you're right, Kiki, I love an airship. I think they're just awesome looking. And every once in a while, there is a sci-fi vision of the future in which these things are just sort of hovering
Or
around
the
city.
past. I feel like a lot of the time it's actually the past. it's like steampunky, right?
Yeah, it's got a very steampunky vibe to it, where we're envisioning a future that maybe would have been envisioned in the 30s or 40s or something like that. Right?
It definitely was an early 1900s kind of thing. But do you remember airship ventures in the Bay Area? It was early. It was during just at the tail end of the early the internet boom. So it was about 2004 or so, or maybe later. Maybe it was 2008. But Airship Ventures was a company that started from Moffett Field outside of NASA Ames, and they launched airships. They had these big, beautiful airships, and they did tours of the Bay Area for about 4 years. And then they failed completely. And I don't know what happened to them. I never heard of the company again. It just disappeared. There was also a company in I think Japan, and there were some others in Europe that were trying to get airships going, or these Zeppelin type craft going for various projects. And I guess the question is, why are we talking about them again? What is happening now in our economy, in our environment, in our climate, that suddenly as this study is certainly proposing, that saying this is a very positive thing. I mean, people have said it for years, but why now? Why again?
Yeah,
Justin doesn't have an answer.
Oh,
No.
no,
He
no.
doesn't.
No. So yeah, the argument is the the renewability, the greenness of it, the not using a carbon emitting source to do the transport of goods or of passengers. Yeah, I mean it is slower, certainly, but if it saves the planet and looks really awesome.
Right. Well,
I
yes.
guess what I don't understand is if one of the main selling points of these is to replace cargo ships and to go across oceans, but one of the issues was that you couldn't predict the weather well before, right? The amount of time it takes to cross an ocean, weather changes, and you end up moving through weather. So what would you do if you were in an airship and you were like, uh-oh, there's weather ahead, and you're over the Pacific Ocean? This is what
There's
I'm not
nothing you can
understanding, right?
do.
Because if you're in an airplane, they're like, oh, we have some weather ahead. Let's take a seat
Well,
and buckle up, folks. And you
No,
have some turbulence,
and
but
it's not.
so
It's not just the Hindenburg that's been destroyed by it also either. There was I'm looking at Wikipedia right now, and there was a De Beers airship. They were using an airship of sorts to go scouting for locations, probably for mining, and it was destroyed over Botswana by a whirlwind. So weather is a huge factor.
Well, but not necessarily. So if we're talking jet stream, we're talking about something that can get to 80,000 feet if it needs to. It can get above storms. Planes will fly above a storm. It's not really usually until they have to land that they're going to hit that turbulence and really have to deal with it in a severe way. So there are, for most of the trip at least, until it has to decide where to set down, it can be immune from the weather effects largely. But it's as much as I would love to see it. It sounds like they're also banking on the hydrogen economy becoming the way forward, which I'm as much as I'm for it, I'm not convinced
That's where it's going to go. Yeah. Exactly. Yeah, I don't know. I mean, it's interesting to see a little resurgence in this idea, and we'll see where it goes and if it actually catches any air.
Huh.
One of the things that might be interesting though is I'm really, because they can carry more cargo, much more cargo than say a helicopter.
Right.
And they can be somewhat stable in positioning. They can sort of position and steer themselves to a location pretty well. I wonder if that would be a good solution in like forest firefighting, to be able to drop a tremendously larger volume of fire suppressant.
Do you really want really, really hot air going up towards your airship?
Yeah, no, I know that could be a problem, but that's helicopters got to deal with this too, right? I mean, anything that's trying to get proper lift over a fire must have some difficulty, but that might be a place to start. Convince California to invest in A Zeppelin firefighting force.
Might be too slow to get the job done, though. All right, moving forward. Okay, I mean, I was looking at stories that were pretty depressing and was thinking we could talk about how human-caused anthropogenic climate change is speeding up the melting of West Antarctica by changing the winds and making the water warmer and melting the ice sheets. We could talk about that, but no. We could talk about the Arctic and talk about how, hey, that's melting, and oh, hey, how fracking in North America is the cause of most of the gas release, the emissions release that we're worried about globally. Hey, good job with that. But I'm not going to talk about that either. What we are going to talk about, I'm going to go back to Antarctica. We're going to talk about dust found in Antarctica. It's not just any old dust.
Antarctic dust.
I
Is
think I
that
lost
snow?
my audio. Do I have
Nope,
my audio?
we hear
you
you.
are.
Good.
You're fine.
Okay, good. For a second, I thought I lost my audio. Dust found in Antarctica, not just any dust, supernova dust. What, you might say? How do you know it's supernova dust? What are you talking about?
Yeah.
Well, yeah, these researchers went to Antarctica and published recently their findings in Physical Review Letters on August 12th. And they melted 1,000 pounds of Antarctic snow. That's a lot of snow being melted. But then they ran it through the mass spectrometer to see what's there. What chemicals are popping up? What masses do they see? What kind of materials might have been there? There's normal Earth materials in there, normal dust components were in there, normal water components were in there, but they also found a bunch of iron and manganese. Now, iron and manganese are normally their molecular variety, their atomic numbers. Iron is iron 56 normally, and manganese here on Earth is manganese 55. It's radioactive, whatever, but the varieties they found were iron 60 and manganese 53. And they went, where did this maybe come from? When we look back in the geologic record, there's iron 60 and manganese 53 that has been timed and associated with supernova eruptions. So these explosions of supernova, and then the supernova creates this massive cloud of dust and particulate matter that if it doesn't burn it all up, it shoots a bunch of it off. And then if it's within our galaxy, as we're tooling around in our orbit around the center of the Milky Way, we can go through that dust cloud of dust from a supernova. And so they're looking at this dust from Antarctica and they run a whole bunch of tests on it to kind of figure out, okay, could this have been nuclear weapon fallout? Could it have been power plant effluent? Could it have been from our own sun? Could it be any of these other things? No, they really, all the molecular evidence suggests we orbited through the remnants of a supernova explosion, but not 2.6 million years ago. Not a long time in the past. 20 years ago.
No.
Wow.
Yes.
I didn't feel anything.
No, you didn't feel anything, but just 20 years ago, we potentially drifted through the dust and gas left over from a supernova. Some of it fell to our planet, was trapped in the ice in Antarctica, found by these geologists, by these researchers. And now they think they'll be able to use what was our position in the sky 20 years ago to be able to triangulate and figure out what star may have gone supernova at some point. So they're going to try and use the timing of it and our potential location to determine where a supernova might have happened. And that'll just give us some really interesting, potentially give us some interesting information about our local environment. Yeah. And so they did it. They ran a whole bunch of mass spec to figure things out, but they think that this supernova would have taken place somewhere within our local interstellar cloud that we've been moving through based on the timing. But
That's really cool.
interesting.
You know, one thing that they might have found if they were looking for it,
What?
microplastics.
Microplastics.
Yeah, so this is
think?
microplastics have been found in seawater, drinking water, animals, going to be in humans too, no doubt. But now that this study they were looking at Greenlandic snow, and I think they were looking at the Alps as well. And they demonstrated that they had higher concentrations of microplastics than they had found anywhere else, with a bit of a caveat. So what this means is and they weren't the same microplastics. So they could actually tell from the different sites that they were testing. And this was a team from the Alfred Wegener Institute, the Helmholtz Center for Polar and Marine Research. They found that these microplastics are being transported over really tremendous distances. And they could tie them to specific things. One of the places they looked at was a rural road in Bavaria, and it was that was the highest hit they got. It was 154,000 particles per liter, and those particular particles were tied to rubbers that were used in a type of rubber used in tires, and it was 14,400 per liter in the Arctic snow, and there was all sorts of things. It was tied to paints, nitrile rubber, which is, I guess, is hard to break down. Different types of paints that could be used for buildings or ships or cars. Sort of interesting though because they've also, when testing seawater in the Arctic, had found really high levels of microplastics, and now they think it could be not transported by the sea, but airborne. Now,
yeah, I mean, dust from the Sahara gets airborne. We know microbes get airborne and transported all around the world. I mean, if plastics
there
are
was
small
another
enough.
study that we're not talking about that was fires in Africa actually are the nutritional source for a lot of Amazonian growth because it gets carried in the wind. So the first thing I thought was, could it be microplastics in the ocean that are evaporating, but they're going up with evaporation and getting into the atmosphere? But this sounds more like it's just not in solution, that the microplastics are just in the air. It is just a form of pollution, which means that we are probably all inundated with them. Now,
I'm breathing them.
The disclaimer part here, the caveat in this study is, even though they have found much higher concentrations of microplastics, their technique is much more advanced. They have an automated system that is being able to do the counting for them, as opposed to physically going in and the way that I guess the researchers have been typically doing it is under a microscope and counting individual particles and manipulating. So without needing to manipulate or lose or count every single thing, they have devised a way to do the counting without human eyes, I guess.
Great. We'll keep counting. Keep counting. No more bean counters. We're gonna have microplastic counters.
Yeah, and they can count down to the size of 11 micrometers too. So they could also the detector can also get down with it's infrared spectroscopy.
That sounds like a job for a robot.
Yeah, very much so.
Yeah, but if they could get down smaller, they may find even more. And that's the part that hasn't really been covered here. It's like they're doing this infrared spectroscopy, and it's 11 microns, but there could be smaller stuff and we're not
Yeah.
seeing it. And and it's the smaller stuff that if you're breathing it in and it's interacting with the cells, we know the shape of molecules like asbestos or this certain molecules in the smoke of cigarettes interact in a negative way with our lungs, leading to cancer and other things. And not lung pollution, air pollution is detrimental to people's health.
And
And
microplastics is now on the list of air pollution, which I feel bad because you left out all the bad stories, and then
here you come.
and I was like, as you listen to them all, I'm like, oh, I should
Ugh.
just kill this story now. Otherwise, it's all
Well,
bad.
let me get you something that is bring us back into some interesting news, just curiosities of our solar system. In May 2019, astronomers noted a burst of energy from the center of our galaxy, the supermassive black hole known as Sagittarius A. It They happened to look at it at the right time. They weren't planning on going to it to know that it was going to brighten up, but they turned their telescope.
Scopes right at the moment in which
Yeah,
it lit
at
up.
the moment in which they were like, whoa, this black hole is putting out more light than it has in a really long time. We caught it on its way back down. So we actually don't know how much energy or how much light, how much brightness the flare actually got to. But what they did see is that there was an increase from its baseline of about a factor of, or actually from the point where they found it, it decreased by a factor of 75
75.
in only a couple of hours. So it was this very intense, very quick burst of light from the event horizon of our supermassive black hole.
You know what this means? Our galaxy has imploded. It's just gonna take a while because that's how things work. It's gonna take a while before the ripple of the explosion hits us, and then it just lights out.
So that's not really how
That's
it
not how that works?
works.
Oh, good.
Yeah, no, this
Whew.
wasn't
I was really
this
hoping it
wasn't
wasn't.
our black hole like just last gasp final moments. That's not what was happening. But we know that that black holes are surrounded, that the edge of black holes is the event horizon, and inside of that we get darkness, right? Around the event horizon there is a disk of dust and gas, and it's all collecting around there. And there are some stars that are very close to the center of our galaxy, and they're whipping around at incredible speeds around that black hole. And eventually they may dive in, kind of like a ball circling a drain. They may dive in at some point, pulled in by the gravitational effect of the Milky Way's black hole. But researchers have a couple of ideas. There's one idea that one of these stars, it's called S2, that it kind of got close to the black hole, and astronomers looked at it a couple of years ago when it was heading toward the black hole, and they expected it to knock some things loose and into the event horizon, which should have been a big light show. But they didn't see anything, and I think they now consider they call it something of a big dud as opposed to a big event. But it could have been a delayed reaction, and so researchers think that this S2 star could have kind of gravitationally moved some stuff that just took a couple of years to reach the edge of the event horizon and to have that energy release and that flare. The other is an object called G2, and nobody knows what it is. According to Phil Plait over at Bad Astronomy over on Sci-Fi, he says at first they thought it was a dust cloud because it gave off a signature of light in the infrared that's kind of similar. It moves around Sagittarius A, the big black hole, on a 260-year orbit. And in 2014, it got pretty close to the black hole. And if it was a dust cloud, it should have been torn apart by the gravitational forces, but it wasn't. So it might be a cloud with a little star in the middle that gravitationally holds everything together and in check. But another idea is that it might have also been a binary star system. And then in these encounters with the black hole caused the stars to merge. And when they merged, they released a lot of energy and a lot of dust. But anyway, if it is shedding material from this dust cloud that this one star or merged binary star system had, that maybe it shed something and a blob of dust fell into the big black hole.
But still,
that's
that
what
seems
they're
like it
doing.
wouldn't by 75 times. You know what I would? My suspicion would be, and we have to ask somebody who would know better. Is what if that's a black hole getting torn apart by a much bigger black hole?
A smaller black hole.
Yeah, like a smaller black hole that has all of that energy condensed into everything that it's pulled in over time. Does it get ripped apart when it gets too close to a much bigger black hole? And does that energy then, some of it get released? Does it gets ripped?
Yeah.
Because if you
Get Phil Plate on the phone. He can tell.
I'll call him up because I want to know what happens if you rip open a
Pick
black
up the
hole with a bigger black hole.
astronomer hotline.
Because I feel like that's what would happen.
Astronomer
I feel like you would
hotline.
get a bit of a fireworks show out of it.
I think there would be an incredible fireworks show, because if it's even a small black hole, it too is going to be surrounded by dust and gas and lots of stuff that, if it's falling in, will, never mind the mass of the black hole itself, but all that stuff around it would be the issue. So I think the big question, though, is it was a very rapid event. It was not a long sustained event. So it was maybe something much smaller than another black hole.
75 times the luminescence of
and that's
the
all we
quasar.
saw.
That seems right, and that was on the way down. That
Yeah.
sounds like something big. I don't buy that could be that doesn't sound like a star or a few stars or 100 stars going in at once. That sounds much bigger to me. I don't know, but we're gonna have to ask somebody as they get
but it's not
further along.
It's not the end of our solar system, not the end of our galaxy. We're all still good. Yeah. Do we have some quick stories for the end of the show?
Yeah, I have a story about Daddy Longlegs. Everybody loves Daddy Longlegs, right?
Like I said at the beginning of the show, I love
know.
them.
Yeah.
So a lot of people I like to see them because I know they eat mosquitoes and stuff like that. But apparently there is an urban legend that daddy longlegs spiders have extremely toxic venom, so toxic that it could kill a human. But the reason they're not dangerous to us is that their fangs are too short to penetrate human skin. You've heard this.
Oh
Yeah,
my
they don't
goodness,
have big enough mouths.
I grew up with this one. Yeah, I know.
More than a decade of research from Lewis and Clark College looked at what was in the venom of daddy longlegs and compiled evidence of the effects of venom on humans. They brought in some tech individuals, some database knowledgeable individuals to kind of make this multidisciplinary to look through the venom and run all these data analytics to figure out exactly what was in there. The long and short of it is, no.
So what's funny is, really, they probably didn't need to do the study. They could have just looked for the research that made this claim in the first place and gone, no, there's nothing there.
Yeah. So there's lots of interesting proteins and peptides. It's highly toxic to insects, but all the evidence they have collected over the past almost decade indicates it has negligible toxicity on mammals. Not just humans, which are pretty big mammals, but mammals, especially compared with black widow venom, for example.
All right, so it's the proteins that are present in the venom. It's not the size of its probably still very tiny mouth.
Yeah, it still could be true that they can't bite, although I don't know about that.
I don't think they're not like very aggressive. Their tendency
They're
seems to
not,
be
but I feel like if you
uh,
like trapped one in your hand, I feel like they would
I've
still
done
try
that
to.
and never been bit. So maybe
I
they're just
don't know.
not
Yeah, maybe
aggressive.
not. I don't know.
Daddy
This is the
Longlegs.
question.
It's
I feel
one
like
thing I'll
Daddy
have to
Longlegs.
unteach my
Yeah.
children someday. I'll get around to fixing that piece of bad information I passed on to them at some point. But we do have a pet pet daddy longleg named Helen, or no, Henrietta. Sorry.
Is she your pet or is she your roommate? Is she
She's
allowed
she's a roommate.
run of the
She's allowed
house?
a corner.
Yeah, there you
But
go.
what's funny is Henrietta's been with us for forever.
You don't even know your roommate's name, Justin.
I know I messed
Jeez.
it up.
How
But
rude.
it's not this. It's not always the same spider. It's just
Yeah, of course not.
yeah.
Yeah.
But
Right now, I have a kitchen spider. It's just a wonderful little house spider, and
Well,
it
name her.
lives, which I love this. It has taken up residence in, you know how the chia pet has evolved, and now it's like whatever you put the chia on. It's this, the clay thing that's shaped different. Anyway, we have a Spider-Man on a
Oh, that's
wall.
amazing. Yeah.
And the little hole that you use to put the water in, there's no chia growing on it at all. It's just Spider-Man and all the spider webs from this spider who has taken up
It's
residence.
Spider's Man.
It is Spider's Man. He is protecting
Aw.
the
That's
little
perfect.
kitchen spider. It's great.
I love it.
And I have to tell I have to tell people who come in. I'm like, you don't clean up that cobweb. Leave it there. She's our friend.
That's awesome.
Yeah. I have a quick story for the end of the show. It's a little earth shaking.
Earth
But,
shaking.
Yeah, because it's about earthquakes.
Let me run for the doorway. Wait, not anymore. That's not a thing anymore.
Wait, that's not the
No.
thing. Wait, why is
No.
that down in the closet?
That's for
Where'd it
late.
go?
Let's talk. Sorry.
We'll talk
we
about
can
it
talk
in the
about
after
that
show.
later.
We'll
But
talk-
I feel like that's the important part. Now I'm going to be
go
ready
under
to
a
really start.
table.
Okay.
Yes.
I need to get a table. Now.
Things that could create a triangular space for you to exist under. Doorways are a little bit more dangerous, but they could still end up creating gaps. If there's nothing else, the doorway is probably better than nothing. That said, a new study in Geophysical Research Letters has looked at a data set that it came up with. If you remember this study from a few months ago, 1.8 million earthquakes in Southern California. There was a new method of analyzing earthquake data that pulled little earthquakes out of the noise of the shaking of the surface of the earth, and it allowed researchers to determine that there were way more earthquakes going on than we had previously thought. Now, a new analysis of this data has been looking at whether or not little earthquakes or foreshocks happen before bigger earthquakes, whether there's something that is predictable in a pattern. And I'm sorry to say that there's no specific pattern per se as to where or how many or how big the foreshocks are before bigger earthquakes take place. But they did find an association with many foreshocks occurring up to 2 weeks before a bigger earthquake in 70% of those larger earthquakes, or it's actually about 75 to 80%. So there's a good 25 or so percent that still can't be explained, or maybe they haven't pulled those foreshocks out of the noise enough yet. Maybe there's more analysis that needs to take place at higher sensitivity. But what they have found is there is a trend for foreshocks to occur before bigger earthquakes happen, which is on the way to creating a path for predicting earthquakes. Slowly but
You
surely.
double caveated. On the way to creating a path.
On the way. Yeah, there we go. We're on the way. We're on the way.
So we're creating the path? No, we're on the
We're
way to
on
creating
the
a path.
way to
We're not-
creating-
For
this is
predictions.
not yet
Yeah.
the path.
Yeah.
For a prediction that
Yeah.
will have a percentage chance of yes.
Yeah.
There you go.
Living in in volcano country up here, Californians in earthquake country, and many other areas of the world who are prone to massive earthquakes, being able to predict them would save thousands
Stay
hundreds
home
of thousands
today.
of lives. Yeah.
Actually, you're in the nexus there in Portland. You've actually got the bigger
Ooh,
earthquake threat too.
Oh yeah.
Yeah, you moved from San
Living in it.
Francisco to Portland. You're just like, earthquakes,
That's I think. where
let
the
me
bigger-
keep them.
I think that's where the
Shake,
biggest
rattle, and
West
roll.
Coast earthquake has happened.
I
was
just
up
don't
in
want to
the-
talk about it anymore, okay?
That's fine.
And then I'm going to be completely human and ignore the impending threat that I can't even contemplate.
It's fine. That's a problem for later.
Hey, what's up, psychology? Yeah. All right. Justin, did you have a last story for the show? Or
Nope. That's it. We got it.
We got it. We have come to the end of our show. Thank you for watching. Thank you for listening. Thank you for being a part of what we're doing. Those of you in the chat room, thank you for being in our chat room, for commenting, for playing nicely with each other in the chat. Yeah, be nice. That's what we want to do. Kindness for all. Let's keep that up. I want to say big thanks to Fada for helping in the chat room and for scheduling our social media posts and writing up show notes. Thank you to Gord McLeod for managing our main chat room. And thank you to Identity4 for recording this show full of the little noises that have been made throughout. Yes, thank you so much. And thank you, absolute thank you to our Patreon sponsors. Thank you to Paul Disney, Richard Onemus, Ed Dyer, Ed, Andy Gross, Stu Pollock, Philip Shane, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Craig Landon, Mark Mazzaros, Jack, Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Dumper 319, Eric Knapp, Richard, Brian Condren, Dave Neighbor, Ben Bignell, Jean Tellier, John Gridley, David Williams, Corinne Benton, Adam LaJoy, Sarah Chavis, Rodney, Tiffany Boyd, John Bertram, Mountain Sloth, Seth O'Gradney, Stefan Albaran, John Ratnaswamy, Dave Friedel, Daryl Myshak, Andrew Swanson, Paul Ronovich, Sue Doster, Dave Wilkinson, Richard Porter, Noodles, Kevin Reardon, Christoph Zuknarek, Ashish Pants, Ulysses Adkins, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Slazewski, Jim DePoe, Greg Riley, Sean Lamb, Ben Rothig, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie Gary S., Robert Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and E.O. Thank you for all of your support on Patreon. If you out there want to become a Patreon sponsor, head over to twist.org. You can find a link on our website. Also, remember that if you love TWIS, you can help us out by telling other people about TWIS. That's right. Get other people to subscribe. That will help us out immensely. On next week's show, we'll be back again, broadcasting live online at 8 p.m. Pacific time at twist.org/live, where you can watch and join our chat room. Don't worry if you can't make it, though, because we have this thing recorded, or at least we try to. It'll be at our YouTube channel, which you can subscribe to, and also at our website, twist.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or just look for it on your phone. Actually, however you found it this time would be a perfectly acceptable way to listen to it next time.
For more information on anything you've heard here today, show notes will be available on our website. That's at www.twist.org, where you can also make comments and start conversations with the host, that's us, or other listeners, that's all of you.
Or you can ignore the other listeners and contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in the subject line. Otherwise, what will happen, Blair?
Oh, your email will be spam filtered into oblivion.
You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
It's all in your head.
This Week in Science. This Week in Science. This Week in Science.
This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views. But I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
Hey, we made it to the after show. What? We're here. We're here. We're here at the after show. We're here. We're here. We're here at the after show. Did we talk about the pulsar glitch? I did not talk about a pulsar glitch. I talked about a black hole thing. Are you talking about the fast radio bursts that have been discovered? What news is there of the pulsar? What have I missed? There were other things. There it is. Astronomers catch a pulsar glitching. They spin faster for only a few seconds. It's like me when I drink coffee. They observed the Vela pulsar glitching. This is a pulsar 1,000 light years away. That's cool. And it will tell us what? By observing the light from it and how it changed, maybe how these things are structured and what's going on, right? Yeah, we can't look deeply into pulsars, so we're stuck on the exterior. These are rapidly spinning neutron stars, very dense. And so, yeah, the glitches, because they slow down. And then, what does it say? The radiation is pointed at Earth intermittently. Oh, they emit electromagnetic radiation from their poles when that radiation is pointed at Earth intermittently. During the star's rotation, we can see the beams. That's the pulsar part of it because it pulses, that light passing like a thing. Sometimes they rotate faster. And when that means the interior of the star is moving toward the exterior of the star. And maybe it just says they're getting some insight. Kind of insight. I want to know. I'm reading quickly. I want to know what kind of insight. What can you see? Okay, the star rapidly spun up before relaxing to glitch speed. That's interesting. The behavior of Vela during the glitch gave them a glimpse into the makeup of the interior of the neutron star. There we go. Neutron stars have 3 distinct layers. One of these components, a soup of superfluid neutrons in the inner layer of the crust, moves outwards first and hits the rigid outer crust of the star, causing it to spin up. Then a second soup of superfluid that moves in the core, catching up to the first, and that causes the spin of the star to slow back down. So you've got these layers pulling past each other. Overshoot is what they call it, apparently. Yeah, pretty cool. Interesting. Thanks for that, Blitz Blots. I did find it, and that is interesting. Immediately before the glitch, we notice that the star seems to slow down its rotation before spinning back up. We actually have no idea why this is, and that's the first time it's ever been seen. All right, back to the models, everyone. They saw it. Now can they model it? Can they make it work in a computer model? Yep. Well, that's exciting. Glitchy. Oh, Noodles, you're welcome. Have I ever had Pamela Gaye on the show? No, I never have had Pamela Gaye on the show. I just met her for the first time in person this last fall. That was great. We've had Fraser Kane before. We should get Pamela on the show. She'd be good. She's a good science talker.
We like that.
Yeah, she's great. Asking about StreamYard, is it free? Yes. So this version of StreamYard that you may see has a little StreamYard logo up in the corner is free. We could try to pay $20 a month, which would get rid of the StreamYard logo and enable some additional branding for ourselves.
Mm-hmm.
And I saw somewhere that we might be able to do videos and various layovers and stuff so that which I'm interested in. I just want to know. I mean,
you wanted
us
to know if you liked it first, for sure.
Yeah, this is the second week. It's working great. Oh, I'll add Justin back. I'll add Justin back to the stream.
What did I do?
You went away, and I was just looking at a plus sign in the screen, and I was like,
Justin, you're so quiet now.
Yeah, it's because I turned down my mic earlier.
I know, but now you're like crazy quiet.
Yeah, it's a thing. It's why I shouldn't have played with the dials. The dials are fine.
You were super loud at the beginning, though.
It was like you could hear distortion.
Yeah, the audio was like you were topping out.
And now you're like,
thank you, Noodles.
Can't hear you.
Yeah, everything's turned down to. Well, this is the new quiet Justin. Just get used to it, people.
Oh, Ed, you're sweet. Yeah, let us make sure that I like StreamYard so far. I just want to make sure that this is really the platform for us. But yeah, I mean, it would be $1 more a month from 20 people. Mm-hmm. One person at $20 a month would pay for it. $5 a month from 4 people would get us the brandable Streamlabs version, the professional Streamlabs Pro version.
$20 a month from one person, $40 a month from half a person.
That's right.
You know, there's lots of ways to do the math.
20
Yes,
people
you
at
split the
$20 a month
people.
also works.
Yeah.
So let's not forget that we could overshoot and still get it.
We need some overshooting. So Blair's computer apparently died.
Yeah, it's deciding to work right now, but my entire keyboard and trackpad wasn't working for several days. That was
Oh.
very fun. And I did some searching, and sometimes very old MacBooks, that just happens, and the computer is dead.
Yeah, I had a computer issue like that. It was a battery. The battery was old and the battery swelled. Because the battery was swelling, that affected the
Oh,
keyboard.
yeah. Wow.
Man.
Yeah.
Yeah,
But
it's time. This is a 2011.
2011.
Yeah.
That's a good old computer.
I bought it before I met you.
You've had this longer than you've
No,
been on
actually,
the show.
yeah, I have had this longer than I've been on the show, but I bought it like, it was very like serendipitous. I think I bought this like a week before I met you because it's a refurb. So it was pretty much a year old when I bought it. But yeah, I bought it like the week before you emailed me and we met for coffee. And then you were like, "Do you have a laptop?" And I was like, "As a matter of fact, I do."
Look at me. I'm gonna sit up 1 second.
Yeah.
Yeah, we have some technology needs coming up here that
Oh, it may be overdue. Yeah, I'm gonna swap out the computer for next week's show and see if that fixes the audio weirdness. that's been
Yeah,
going on.
so somebody made some comment about an audio Windows issue earlier. Like there's a lot of different tumescent battery. Thanks, strengths. Yeah. Some people have said maybe it is a connection, maybe it's a cord going bad. So if we can swap out your cords, your cables, it could be I don't know if it's a plug, like a power cable plug
That's
going
fine. I mean,
bad. Someone else said maybe it's your sound card.
there's a
Yeah,
lot
Identity 4.
of cables.
Identity 4 said maybe USB driver issues, which that might be. You have a PC and drivers sometimes. Maybe you have a driver that needs to be updated.
Could be a lot of things.
I had a weird sound like this a long time ago, and I remember it. I think it ended up being fixed with a driver update a long time ago, which was really exciting for me because I thought I was going to have to replace everything.
I never let anything
It was a
update
long
on
time
this
ago.
computer, just because I feel like when you update things, that's when they go wonky. So I don't know. I'll swap out the entire computer and see if that has an effect, and if so, we've narrowed it down to the computer and not a cable.
Right.
If
Okay.
that's not a thing, then I'll start replacing cables.
Oh, look at you thinking scientifically.
It's kind of a thing.
Man, I would go cables first, but that's just me.
Well, I already have the other computer.
Oh,
I
okay.
don't have the other. I'm being
I
cheap.
see, I
is
see, I
also
see.
part
I
of
see.
it.
And there's more cables
taking the
than there are
convenience.
computers. Also, yeah.
Oh, there's a lot of this.
Yeah.
It wasn't until like you were saying like wiggle your cable, and I'm like, which one? Is there power next to any of the cable? Oh yeah, there's power cords and cable cords and audio, and it's going from this thing to that thing, and then back into it. Maybe the cable that's going back into the same thing might be a problem. I don't know. Is the thing. But yeah, I'll try the big thing first, and then we'll whittle it down.
Yeah. USB port could be going out, says Identity 4. Yeah, computer first. Let's see if it's a computer problem.
Yep.
That'll be great. And then we'll go from there, and hopefully we can get this fixed, because this is it's a terrible noise to edit out.
It's awful. And it's been 3 weeks, 4 weeks.
Yeah, and it's gotten worse. It happens more often. It happens more often now than it did.
I wonder if it's the heat in the garage. It's causing problems.
So it happened on
Maybe.
Google Hangouts. It was before we went to StreamYard.
Yeah.
Yeah.
Okay.
Yeah, it was omnipresent. I think the last couple weeks of it.
Definitely last week. I mean, last week was the week that we were like, hey, that's weird.
Yeah,
Mhm.
Yeah.
and it's doing it. So I actually can't hear if and when it's happening because my audio doesn't feed back to my headphones. So it's also it's I can't like self-diagnose it. I would also need
No.
And we did. I think Blair, before you got here, we did a bunch of cable jiggling. Nothing seemed to recreate. It seemed to be when I was sitting still and randomly it would happen.
Yeah. Random. It's not a pulsar glitch. It's just in somewhere in your system glitch. Identity 4 says last week was the first time he noticed it. Yeah.
I mean, it does seem that this system is more nuanced.
A little bit, yeah. I just don't. I
So
mean, it's browser based,
something
so I
else.
don't know why that would
Can I tell you something weird that I
cause
don't
it.
understand?
So it could just be correlation.
When there's also a weird thing here when I mute, which I just did there, and the two of you are speaking. Especially, I hear it with Blair. Your S's become slightly digitized. They can become lower resolution when when my mic when I'm unmuted, and when I mute, your audio quality goes up a little bit. It's a weird
Interesting.
thing. Yeah.
Maybe it's your core. It's your
Sound
headphone
card.
cord. Oh yeah, or it could be your sound card. Yeah.
Or it could be a port, or it could be a driver. It could be, that's why we're doing this. New computer, well, not new, but a different computer next week, and then we'll just see. And I think actually that one's a better quality one too. Come to think of it, I don't know why this one ended up being the workhorse.
That's funny.
Hot Rod says that you can go into sound properties, and on the listen tab under microphone, you can hear yourself, and then you can move stuff around and see if you can
Sound
hear it.
properties.
Yeah.
I don't see it on this. I mean, I can do yes. Outside of the settings of this app. Okay, gotcha.
Ooh.
I don't know.
I don't know. Does anybody have any travel or anything coming up that's exciting?
I'm going to New Orleans
When are you going to New Orleans?
in September for the Association of Zoos and Aquariums annual
Right.
conference.
Right. When is that?
That is, I leave September 7th, which I think is a Saturday, and I get back on the 17th. I am hoping to do the show,
It's a week.
on the Wednesday, on the 11th. That is my plan. It depends on the Wi-Fi at the Airbnb,
Yeah.
but
Okay.
it's my plan.
All right. Airbnb
Is there
Wi-Fi.
something interesting on the radar for a thing we might be able to do?
What?
Or are you just checking to
Oh,
see if
no.
anybody's going to be dropping out in the near future?
I'm just checking. When are you gonna be here? What are we gonna do?
In October, in early October, I will
Uh-huh.
be in Churchill teaching a climate change communication course to polar bear scientists,
Oh.
polar bear researchers, not
Ooh.
polar bears that are scientists, but scientists who study polar bears.
Oh, okay. All right.
Um,
I was like,
good.
And I
didn't
do
know.
believe, I have to check the
Very
schedule, but I
dangerous.
think Wednesday night is the night that we are staying in converted train cars in the middle of the Arctic with no phone and no internet and nothing.
Oh,
Wait,
that's
you're
gonna be
going to the
awesome.
Arctic?
Yeah,
You're
I
going
mean,
to the you're going
I'm
to
going-
the Arctic.
yes.
Like Alaska?
Yes. So Churchill, and then taking these buggies out for like
Wow.
hours. So
Awesome.
That sounds awesome.
You're going to have to bring a camera.
good idea.
Oh.
Like when you can like report
Yeah.
from the field and
Yeah. No, actually they asked me
do a
if
short.
I wanted to do any twist shorts or anything like that while I was there, actually.
Yeah,
So
perfect.
I will be bringing some sound equipment with me if I can manage it.
Awesome.
Yeah,
And you're going when?
October.
Okay, so that's kind of wintry.
It's
Yeah,
actually
well,
it's
no.
one of the better times of year to go.
Okay, cool.
Interesting. Where is Churchill? Is this Canada?
Yes, it is the peak of Manitoba. So it's on
Manitoba.
Hudson
Okay,
Bay.
here we
I'm
go.
looking at it right now. So it's
not.
I don't know. It's not technically. Where is the Arctic?
CA,
It
NA,
might not technically be in the Arctic.
DA. I'm going to put a picture of Churchill's.
Oh, Canada's just north of the United States. I didn't know that.
Yeah.
Look at
Ha!
that. I had no idea.
You're so silly sometimes. So
I thought it was
there's
next to Finland.
right now it's mostly cloudy and 51 degrees Fahrenheit,
Mm.
12:23 a.m. and probably still quite bright.
Mm-hmm. Mm-hmm.
Probably. So, okay, so you're in this little tiny place and then you're going to drive little tiny cold place and then you're going to drive places. Do you know where you're
Yeah.
going specifically?
Out looking for polar bears.
The Churchill Northern Studies Center.
Just out chumming the ice.
That is where we will be. And then I'm trying to look. And so we're slightly south of the Arctic. Hudson Bay's right on the tip of the Arctic Circle. So we're not quite at the Arctic, but we're very far north. So we're going to be basically going out in these buggies almost every day. We're going dog sledding one day, which is very exciting. But almost every day we're going out, just spending hours searching for polar bears and doing our learning on the buggies. And then we're also doing learning like over breakfast and over drinks and dinner. And but then there's one point when we're venturing very far north and staying in converted train cars that is now a research center.
Oh, that sounds
there's
like so
this-
much fun.
Is it the Churchill Northern Studies Center?
So that's our home base.
That is your
I
home base.
believe.
Oh, okay.
But the other thing is like slightly more remote, and I think that's where we're going to be on Wednesday night. So-
Tundra Buggy Lodge.
Yes,
There it is.
Yep.
Tundra Buggy Lodge at Polar Bear Point.
Yes.
Oh, here we go. So
That's a good
wow,
place
you
to
are
look for polar bears.
You're going way out there. Let's look at some pictures of this. Are you going to get to see the Northern Lights?
I don't know if the timing is right. I hope
Look at
so.
that. Look at those.
Yeah.
Snow train.
Yeah, it's
Look at
still
this.
piercer.
Oh, look
It's
at that
so beautiful.
build. Oh, that's
It's-
nice.
Beautiful.
Yeah.
Look at that.
Converted
It's
train car.
gorgeous.
But yeah, there's
It
no
is.
cell reception. There is sometimes Wi-Fi, but it is not dependable. So I will try to I'm supposed to be part of my job while I'm there is to be active on social media. Like that's part of the exercise. So I will be tweeting and Instagramming and Facebooking all of my adventures. But I will also try to pop in the chat room depending on how things are going. If there's Wi-Fi, I don't know. You can send me the link and I can try to pop in for a second, but there's a good
If
chance.
there's anything, yeah.
There might
yeah.
not be anything while
You
we're
know, a good way to
You
spot polar
definitely
bears in
need
the
to
tundra.
record some video.
there. Yes.
The best way to find polar bears in the tundra
How? Strap
an
garbage on yourself?
with an airship.
Oh, with an airship.
Yeah.
Yeah. So, Kiki, I think I'm gonna break down and buy a selfie stick
Get it. Do it.
because
yes.
I have all of the equipment to hook to record sound through my phone. So I think I'm gonna get a selfie stick and try to record video.
Oh my god, I'm so
Maybe-
jealous.
maybe
this
use it
looks
for a couple selfies.
amazing. This looks ridiculously amazing. I'm so jealous.
I know, I'm pretty excited. Like, oh, yeah. So I got I'm friendly with and I've I've collaborated with a woman who works for Polar Bears International, and she sent out this email to a bunch of people, and asking if they wanted to take this course. And it's a course in the Noki leadership, which I did last year. And I replied, I was like, so I can't take the course because I already took it, but if you need teachers, let me know. And she replied almost immediately and was like, actually, we are looking for teachers and your name already came up. So good to know you're
Nice.
interested.
That's fantastic.
Right.
So very fun. I'm very excited.
Yeah.
I am too.
Oh my God. I don't even know what I'm going to pack. It's going to be insane. They provide some of the stuff. They provide the boots when you go out in the snow. They provide the giant parkas.
Good.
So they're kind of
You're
just like,
still gonna want
dress
your
like-
own jacket.
just dress like, it's kind of cold. We'll take care of the rest. It's
I wonder what the temperature range actually
Wow.
is going to be, though. If you've got the, that was one of the weird things when I was in Greenland, is that it was around 40 degrees the whole time,
Not that far.
because it was daytime all the time. So
Yeah.
if you're there in a near never ending sun, it might not be super cold.
so October, it says Churchill, the average high is 43 and the average low is 30. So it's actually not that cold.
Yeah,
Not
it's
too
a
terrible,
pretty tight
yeah.
range. Yeah.
Mhm.
Awesome. That is so cool, Blair. That's amazing.
Really excited.
So it's on Hudson Bay. Okay, interesting. Wow, that is a long way away from here.
So I'm flying to Winnipeg
Yeah.
and we're meeting and doing icebreakers and initial stuff in Winnipeg. And then we are all taking a teeny tiny plane together to Churchill.
And this is what, like a 2-week long trip or something?
Well, so I leave on the 27th or the 28th. I don't remember. And then we get back to Winnipeg on the 5th, I think. And then so it's just a week. And
That's faster. Yeah.
then my mom said she wanted to come out, so she's going to meet me in Winnipeg. And we're going to
Cool.
mess around in Winnipeg for a few days before I head back.
Awesome.
Perfect. Oh my gosh. What
Yeah.
an adventure. That's going to
Yeah.
be, that's an adventure. That I cannot wait to hear about that.
Yeah, I'm pretty excited.
Holy moly.
So it says, let's see, in August in Churchill, sunrise is at 5:30 a.m. and sunset is at 9:04 p.m.
Eric in Alaska is, he's well aware of this. He'll be in the dark. You'll be in the dark in October.
Neat.
Mm.
A lot of dark.
Very- Let's see, October 7th.
Yeah. So 2 things: don't get eaten by a bear,
I won't.
and
That's both of them.
don't go walking on thin ice.
Oh, I
yeah.
definitely won't do either of those. Don't worry.
I have good advice for you. There we go.
That's
I can
good.
help.
And everyone in the chat room telling me not to get eaten by bears. Remember, I was a bear keeper. I
Yeah,
know
that's actually the
nowhere
one thing
near
I'm-
bears.
That's the one thing I'm not worried about, is you getting too close to the
no.
the bears.
Yeah, it's
Yeah.
like the people who work with the animals
Oh.
usually are way more cautious of getting too
Yeah.
close than the people who have not worked with them.
hmm
Yeah.
Bears are cute.
Yeah.
I got my finger licked by a bear.
Oh my God. What kind? Black bear?
No. Brown bear?
Grizzly bear?
No. I don't. Might have been grizzly.
I bet it was a black bear because they're sometimes brown.
Yeah, I don't remember it. But it was in Washington at University of Washington research facility where they had captive bears that they were studying.
Yeah, it could have been a
And they were
grizzly
doing-
bear.
Yeah,
Could have been a black bear.
yeah. But it was very nice, and it sniffed my finger, and its long tongue came out and licked my finger.
That's awesome. I'm
So
glad you
cute.
still have your fingertip. That's good.
Yeah. I didn't put my hand in the cage. I put my up to the edge of the cage. It was all fine. Totally fine. They weren't zoo bears. These were wild bears that were in captivity, but they were very sweet. I mean, not that I would have gone in, like I said, I
Yeah.
wouldn't have gone
Yeah,
in.
they're
with
real sweet
them.
till you're in with them.
Yeah.
Yeah, there's a whole generation though, of which I'm at the tail end of, or maybe they were just reruns back in the rerun TV days. But one of my favorite shows I remember as a kid when it come on was Grizzly Adams,
Oh yeah.
and it was just like Mountain Man and his friend Bear,
Yeah.
and they'd go around and get into trouble, and then the bear would help him.
No.
Bear would help him out of trouble because it was like his dog,
I mean,
basically.
There's that. There's Tarzan. What's the difference?
Yeah,
None of it's smart.
yeah.
None of it is smart.
Yeah.
Don't do dumb things with wild animals, people.
Unless you're a mountain man, in which case that's part of what.
No, if you're a mountain man, still don't do dumb things with wild animals.
Mountain men
Well, they would
usually
know better.
know
Yeah, they
more
know
than
better.
any of us.
Otherwise, they're not a mountain man very long.
Yeah.
They're a mountain meal pretty quickly.
Mm.
Okay. Is that it?
Yeah.
Is it a wrap?
Yeah, I think
right.
that's it. Yeah, I don't have anything going on. School's coming back into session in a couple of weeks. Oh my goodness. Back to the normal weekly grind. I'm not ready. I'm not ready. Oh, yeah, if anybody wants to meet me in Las Vegas over Labor Day weekend, I guess it's soon now. I am going to be at a conference called Biohack the Planet, and it is organized by Joshua Zahner, who is the biohacking DIY bio guy.
Josiah?
Josiah,
Zahner. Yeah.
who we interviewed. He's the one who did the fecal micro.
Oh yeah, no, we
transplant.
tell that story all the time. I always find it really fascinating. Yeah.
Yeah, so
The
I'm
cookies,
going to.
the sugar
The sweet
cookies
tooth.
Yes.
Yeah.
that
Yeah.
that came from his microbiome forced him to buy a thing, and then
Oh.
days later, he figured out why.
Yeah, and this week there's a study out where they looked at the microbiome of gosh, if I can find it. It was the microbiome. I think it was the microbiome of the tongue of obese people, normal weight people, and underweight people, and they found differences. I think that's what it was microbiome, and so I thought, oh, that Justin might be interested in that. See if I can find that story. It was just like this week. Oh. Gut microbiota influences insulin sensitivity, but that's not what it was. Oh, I wish I could find it because you'd be interested. But anyway, the first thing I thought of was Josiah and his study and what had happened with that. But I don't know where I can find it.
Something ominous. Eric in Alaska was just saying the thing you gotta watch out for in bears is the mood swing, and then he disconnected.
Then he's
right
like,
as he's giving the bear warning, he was gone. I don't know.
Man.
Again, let me just reiterate. I got it.
Okay. All right. In that case, I will sleep
Yeah.
easy. Say goodnight, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin. Good
night,
Kiki.
Good night, everyone. Thank you for joining us for another show. Hey, co-hosts, thank for a wonderful show tonight. It was super fun. We'll get these audio issues and computer things and streaming things all worked out in due time. But it was a great show. Thank you. Okay, how do I make this stop? End broadcast. And then again, end broadcast.
Still
There's so
going.
many buttons.
I'm still
There's
on
other
the